So this project is about automatic blood cell image identification and classification using a MATLAB programming software. Here what we are trying to do is basically depending upon the cell shapes and their sizes, we are classifying them in an image. Now basically blood has multiple cellular components and a plasma. Now each cellular component's count is very important in the view of diagnostic and as well as to identify what is the problem with the patient. For example, if number of WBCs are high, that means that patient has infection. If the number of platelets are high or low, depending upon if the platelet goes low, then the person may have dengue. If it is high, it could be a platelet cancer or something like that. So depending upon what kind of what is the count. Now there is a large range of count variation, but once it exceeds, then there is a problem. So most of the cases, CBC is like a window through which we can see through into the body and check what is the status of the body. Just by prick of finger we can do that. So here in this picture what we can see is biconcave shaped RBCs, different SEM images of blood cells in which you have neutrophils, eosinophils, basophils, the name given by neutral, acidic and basic. Then lymphocytes, which are classified further as B and T lymphocytes, and monocytes. Monocytes are the bigger cells and they are the first fighting mechanism of the blood. Platelets, which are small parts of a large cell, and that's why they are parts of, that's why the name platelets. And erythrocytes, which are more popularly known as RBCs, which overrides most of the count because they are almost 10 to 100 times than other cells, and that's why our blood color is also red, because most of the cells are red blood cells. Here we have another WBC picture and some SEM images, falsely colored, pseudo colored images. What is missing on this slide is references, if we have taken these images from which website or something. Now with this project, what we are going to do is provide a valuable information to the pathologist for their treatment. So what we are doing, we are trying to replace an expert who can see on a microscope and tell us what is the problem in the blood. We are taking a software which will be cross verified with the expert's information, and depending upon the corrections he suggested, we will set the values in the software so that most of the expertise can be made available in the remote areas, villages, everywhere, where the expert need not have to be present all the time. He can just tell us what is the problem with the blood, and that problem will be fed into the software. This will obviously save a cost, a manpower, and most of the times if the person is new who don't have enough experience, he can make mistakes, but a software trained with the expert with that kind of knowledge, software will make less errors. And in medical conditions you don't prefer any error, so generally all the medical equipments, they are acceptable, will have accuracy above 99% only. Now what is blood test? So basically CBC, when we go to the pathology lab, we give out a blood which is minimum quantity they take out, which is around 2 ml, and even your machine like Sysmex takes around 25 microliter of blood, but they have to take 2 ml because they are taking it from venous blood and they can carry multiple tests, a repetitive test, so that they take more amount of blood. Few times it happens that blood gets clot, and that's why a few of the blood cells are wasted, so that's why the extra amount is always taken, because in handling it might damage. So generally they are taken in 2 tubes. Those tubes are EDTA coated to avoid the coagulation, and they are of 2 ml tubes where they collect the blood. There are other tubes where there is a predefined platelet RBC lysing solutions or WBC lysing solutions for the other counts. So RBC count lysing solution is used to remove any WBC or platelet counting mechanism, so that RBCs are removed in RBC lysing solution and WBCs can be easily counted. So that's why they put in the tubes. It cost ranges from as low as 100 rupees with all discount, and it can go up to 800 rupees also, depending upon what machines you have. Most of the cases, this 180 rupees average test what we get is a manual test. Most of the machine test, because machine needs reagents, the test is costly. So what I was talking about, a machinized test, what we call a fax test. This is basically a fluorescence activated method where they sort the cells and then count. The sorting is done so that most of the fluorescence tag will be to the WBCs, to the nucleus, and RBCs are already outnumbering the entire solution. So tagged WBCs can be easily found out with the fluorescence reflectance as well as platelets, and then depending upon positive or negative voltage applied at the plate, the cells changes their path in the flow, and that's how they get sorted, and further they are counted through either a optical method or a electrical method. Electrical method uses Coulter counter method principle, where each cell is passed through a hole and then counted, whereas in fax each time light gets cut through, how much time it was cut through and what is the power at which it was cut through can determine what is the type of the cell. So this is electrical techniques. What we have, impedance measurement, where a cell passes through two electrodes, gives a pulse, and the amplitude of the pulse is corresponding to the size of the cells, and width of the pulse also determines the cell also has three dimensions. So it's like width of the amplitude of the pulse gives you height related information, and pulse duration gives you 2D XY zone area of the cell. So this is how they determine. More or less we assume the cell to be a sphere, and that's why we consider the width as more important point than the height, because amplitude may vary with the noise but width cannot vary with the noise. So that's why electrical techniques are more precise as compared to other techniques. In flow cytometry we have two detectors, one scattering detector and one forward detector. So scattered light is detected by a secondary side scatter detector. Depending upon what kind of cells, the scattering will vary, and then depending upon the signal received by the scatter detector will be considered as type of the cells. So this is these are the basic principles that they are used in this. There is photospectrometry, where a light is passed through the cell, and this is not very useful for blood, because blood mostly have a red color and red spectrum goes down rapidly, and spectrometry is not considered as good as Coulter counter and fax. So spectrometry is a old technique which they used to use previously, but nowadays nobody uses this spectrometry technique because it was not so accurate. So other than spectrometry, they need some low cost methods. So there is a hemocytometer, also known as Neubar chamber, which has a fixed size grid. A Neubar chamber is raised to a height where it can fit only 10 microliter of blood when you put a cover slip onto a Neubar chamber, and also its height is so precisely adjusted so that only one cell lies at a time, so that there is no overlap of cells on a focusing plane, and that focusing plane is further divided into specific grids. These grids allows us to calculate the volume. Generally each grid will have 1 nanoliter of volume, depending upon what grid size I am talking about. But that 1 nanoliter, if it has 270 cells, then what is the amount of cells in 1 microliter? It back calculated, and multiple times calculation is done so that to avoid the error. So each Neubar chamber has two symmetrical chambers, so that the count can be doubled or counted 2 times. Further, it has 16 sub blocks and then 25 sub blocks, and then out of which 5 diagonal blocks we choose. The limitation of the process, I don't think this slide fits here, too expensive, but I guess it can be other active processes, so this slide doesn't fit over here. Yeah, so still prone to errors, and fax doesn't have that much of error, only 1%, and it needs a human, because at least fax needs the agents, 3 agents to be replaced, as well as Coulter counter needs your sample loading and all, but that is required in our project also, and blood, yes, quantity is more, where we can work with a fingerprint. The main advantage of hemocytometer over the automated techniques is hemocytometer takes very less amount of blood, which you can easily take from a fingerprint. It doesn't require any electricity, any kind of mechanism other than optical microscope, and since our algorithm is running on a computer, we can use laptop as we talk about electricity requirements, and also time consuming, and here we can actually see what is inside the cells. So trusting a machine and seeing it by eye makes a lot of difference. So our motivation of the project is to consider not to replace entirely automated counting system, but to give some better solution to a manual counting process with kind of expertise that we can get, and also, so basically save power, save cost, and replace the human efforts, that is expertise requirements are reduced. That is the motivation of the project. We still need to add few papers in this, minimum 10 papers. So this is not about literature survey, but what we need. So it goes under a methodology. We need a microscope with camera interfacing. Syringe we don't need actually. We need something like a finger pricker, or a syringe can be used to prick needle. Then we need a FEDTA anticoagulant, but generally if you take prick and within half an hour if you finish all the process, you don't need EDTA also, because after pricking also there is some time taken by the coagulation mechanism of platelets to happen, which actually traps the RBC, and Rolex formation is also avoid of RBC if you do it immediately. So that is a point. So we are replacing or removing syringe and anticoagulant. Then also diluting fluid we might require, but tuber chamber is designed in such a way that diluting fluid is also not required. And we need of course a running software, which could be a sub EXE generated out of MATLAB, so that user need not pay every time for a MATLAB, and once the code is ready we can just distribute the EXE with the permission of MATLAB. Now steps we are going to follow in our projects are, we will start with a blood, which may not be 2 ml, it can be 10 microliter also. Then mixing it with diluting fluid is not necessary step, it's optional step. Fill the chamber with hemocytometer. For that we need pipettes. Capture an image through a microscope, store the image and start with the MATLAB processing. Give an output count and end. So this is a flow graph or block graph for the entire project, but where my work comes is under this MATLAB programming. So I have to redo this bit. Now inside hemocytometer, so this slide should be there where I explain hemocytometer. So inside hemocytometer, like I was saying, there are 9 major blocks. Inside there are 16 blocks on 4 corners for WBC count. There are further 25 blocks inside a central chamber, which is further divided as 3 lines and 16 sub blocks. Now we have to count for each 16 sub blocks, and out of 25 we can count any 5. So 5 into 16 sub blocks, and then depending upon what is the count, either we can average out, or there is a formula of multiplying factor, like each sub block can hold up to I guess it's 0.001 microliter, and then we have to multiply it with the corresponding number of cells. So in 25 blocks, if you have 100, then 100 cells in 0.001 microliter, then correspondingly what is the cells per microliter. So there is a multiplying factor straight away. Now blood cell classifications, this slide should come where you have given those diagrams like erythrocyte, leukocyte, thrombocyte. So these two slides are really relevant. Now after literature, you always start with methodology. So how you are going to do, so your software algorithm should start after this major block diagram, which is I guess here. So this should be the next slide, and all these two should go where you talk about the cells. So in male and female you have this different ranges, and but the ranges generally overlap. Always WBC and platelet also has a range which also overlaps. So in software, what we do, so this should not be like this, it should be a flow chart properly drawn. So we start, we will do a image acquisition through a camera. Here you should mention what image acquisition. So each block should now become a slide. So for image acquisition, you are using CCD, you should explain about CCD. So inside image processing, what you are going to do, what is pre-processing step? So we are going to apply median filter to remove salt and pepper noise. We are going to, which is generally generated in CCD cameras. Also we are going to apply a Gaussian smoothing filter with very noise removal if required. And so basically image pre-filtering and contrast adjustment are the two steps we are going to do. Once we do that, we are going to extract features. Inside that there should be a sub blocks which tells us about shape extraction, then edge detection mechanism which we use, then neighborhood pixel processing, connected neighbors, and that segmentation part goes under that. So image segmentation, how we are going to segment the image. So read about that rice grain wala MATLAB ka theory. Every steps they have mentioned should come here. And then classification. So classification basically based on Euclidean distance. So clustering, how we are going to do minimum distance decoding. All these things fits under a classification, and then the output. So it goes back if there are further images, then or it reaches end. So this has to be redrawn with proper flow chart kind of thing. Now advantages of our project. So these should be two separate slides with photograph. Not much expensive. What we can write is cost effective, comparatively fast, not exactly, but yes, resource limited settings it can be used, prone to less errors, yes, because it's a software as compared to manual counting methods. You are not challenging the Sysmax or Coulter counter electrical methods. Resources needed cannot be a disadvantage, but here you can say it highly dependent on a camera based microscope. So if you have regular microscope, at least you need to have a CCD camera, and also you need to have a one processing machine, which is generally now a giveaway thing. So any Pentium 1 to Pentium 2 or even i7 processors you can use. So depending upon what is your budget, you can purchase a computer and run the code. Our aim, not my aim, give a cell count with the help of a MATLAB. Here we can give multiple aims. You just don't want to do that. We have to classify the cells, give out the RBC count, give out the WBC counts, give out the platelet counts, and if possible, give out the differential WBC counts. Also, it can able to give you something like shapes and size based distributions, histograms, and these are the few references. That's it for this slide.